Thanks to all people for connecting to this seminar of Sapienza Neuroscience, organized in Sapienza University of Rome. The speaker today is Professor Simone Rossi. He's a professor of physiology at the University of Siena, Italy. He is the leader of the work group of International Federation of Clinical Neurophysiology on the guidelines for the use of transcranial magnetic stimulation in clinical neurophysiology. But he's also a pioneer of the use of transcranial magnetic stimulation in cognitive neuroscience to investigate higher human functions such as episodic memory or reasoning and many others cognitive functions. And he probably will show us also some really promising therapeutical perspectives of these procedures. So I kindly ask Professor Simone Rossi to start with his seminar.
Thank you very much, Claudio, for this kind invitation, and good morning to all the virtual attendees. It's really an honor for me to give a seminar here at La Sapienza. And the title of my talk is The Electric Oscillatory Brain and the Challenges of Neuromodulation. That are topics that you could find eventually also in this book that I recently published for Raffaello Cortina Editore in Milano. Of course, the book is in Italian, not in English. I will talk basically of techniques of non-invasive brain stimulation and try to explain how they could work, could interact with our brain activity. And then I will give you some example of how non-invasive brain stimulation can be used in the context of neuroimaging investigation to investigate brain functions, high cognitive functions. Then a few words about how non-invasive brain stimulation techniques can use as a neuromodulatory technique for therapeutic applications in certain psychiatric and neurological disorders. And a final part will be devoted to less traditional neuromodulatory use of non-invasive brain stimulation in Alzheimer's disease and patients with brain tumors. But this is still at an exploratory phase. So for who is not familiar with brain stimulation, let's start with probably the most known of this technique, which is transcranial magnetic stimulation, in which a coil placed on the scalp surface, into the coil is flowing a time-varying, very high currents in the order of thousands of volts. And this creates a magnetic field that can penetrate painlessly inside the brain, where, for the Faraday's principle, it induced a current into the superficial layer of the cortex, which is able to stimulate surface cortical neurons. This kind of stimulation, by using neuronavigation, can be precisely guided on the target region that we want to stimulate. So it's a very precise way to stimulate some neural assembly in the surface of the cortex. When it was introduced in the 1985, transcranial magnetic stimulation opened the way to investigate non-invasively in humans the corticospinal system, from the cortex to the target muscles. And it opened a totally unexpected perspective to investigate human motor functions. But we will not talk about this possibility of using TMS. What we are interested in this context, in the context of neuroimaging, is that this transsynaptic activation of superficial cortical neurons can produce, besides muscle twitches when applied on the motor cortex, also changes in underlying oscillatory activity in the brain, but also changes in blood flow and metabolism that can be picked up by traditional neuroimaging investigations. And last but not least, when TMS is repeated several times in certain pattern of stimulation, can even modify behavior. We can use different type of coils. We can be very focal in the stimulation or less focal for particular clinical activity. And of course, we have to take into account, in order to stimulate at the best our cortical target, that the intensity of the stimulation decreases with inversely with the square of the distance from the cortex to the scalp. So the intensity of stimulation should be adjusted in a personalized way to the subjects or patients under investigation. Let's start the first topic of my talk, trying to understand how TMS can fill some open gap of the current neuroimaging investigations. When we want to study the brain, we have several techniques available. Of course, we have structural MRI and even tractography, where we can see axons traveling through different regions of the brain. So with the structural MRI, we can have wonderful anatomical informations, wonderful details of the lesions, but nothing regarding the function of the brain. Some of the functions of the brain can be explored by functional MRI, either in case of bold acquisitions or placing the subject for 5 minutes into the scan and looking at the resting state activity. In this way, we can obtain wonderful spatial resolution imaging. In this case, this brain is looking at probably at some visual stimulus, because visual cortex is primarily activated. And we can also investigate how the different activated regions, either during a task or during resting state, are connected each other. So we can approach human connectome to understand better. But there are some constraints in terms of timing resolution, because in order to pick up this blood oxygenation related signals, a long time is necessary, around 500, 800 milliseconds. And 800 milliseconds for the working brain is a sort of eternity, as you can imagine. By studying oscillatory activity, electrical oscillatory activity in the brain, with the different brain rhythms that in different regions, and how this rhythm can couple or decouple in terms of oscillations, we can obtain a great temporal resolution in the order of few milliseconds, even less, Claudio would say. And of course, we can also investigate connection, how these two distant regions or two close regions can speak, exchanging these electrical signals each other. But the main problems of both functional MRI and electroencephalography is that the information that we can gather is not causal, is a sort, cannot give any answer regarding the real necessity of this activity, of this electrical activity in the task under investigation. And this can be done with brain stimulation by targeting a node, for example, of a network, of a complex network with TMS. And if the stimulation, the interference with TMS with this node can produce an error in the task under investigation, then we have closed the loop with the other neuroimaging techniques. So we can choose the region to stimulate on the basis of previous neuroimaging studies for the task under investigation, and then we can prove with TMS if that region is really participating actively in the task by inducing errors, either in accuracy or in variation of reaction times. So in order to get a good interference with TMS, we have to fulfill three constraints. We should know a priori the regions that have to be interfered with TMS, that are our targets, on the basis of previous studies. We should reach a good selectivity of the stimulation for the brain region that we wanted to be interfered with, of course, and there should be a strict temporal link between the interference in the neural process under investigation induced by TMS and the task. And this is what we did together with Claudio Babiloni, unfortunately, almost 20 years ago, but at the time it was really innovative. We decided to approach our episodic memory functioning by using TMS, and we were a pioneer in this sense at the time. At the end of the last century, many neuroimaging investigations on memory disclosed that our prefrontal cortex, both left and right prefrontal cortices, were deeply involved either during the encoding of memoranda or during the voluntary retrieval of memoranda. And Tulving at the time conceptualized these neuroimaging investigations, introducing the HERA model. That means hemispheric encoding retrieval asymmetries. Healthy subjects showed a left prevalence activation in the prefrontal cortex during encoding tasks, and a right prefrontal prevalence activation during the retrieval task. But again, this information were of course very nice to see, but this kind of activations did not tell us anything about the real necessity of these activations for a successful memory completion. They could be simply epiphenomena, or they could be, for example, assemblies of inhibitory neurons activated that inhibited the task. So we could not have any answer in this sense. So we thought to test this episodic memory with TMS. And we applied a standardized task of visospatial episodic memory in which I'm here some voices, there is some microphone that is
compared to the other, you need to be close for a certain amount of time before it starts speaking.
So we use this task in which during the encoding phase, we showed the subjects some pictures that could be interior of apartments or external landscape. And some hours later, in the retrieval task, we presented a new set of pictures that could be again already seen during encoding or new interior pictures, and these are distractors. During this pictures presentation, testing the memory function, we applied a train of repetitive transcranial magnetic stimulation in coincidence with the first time of pictures presentation. What we found is that when the left dorsolateral prefrontal cortex was interfered with TMS in encoding, the number of hits dramatically decreased versus all the remaining conditions. And the same happened when the retrieval phase was interfered with TMS on the right dorsolateral prefrontal cortex. All the other conditions, right stimulation in encoding, placebo control condition, left stimulation in retrieval, and no stimulation, did not affect the memory accuracy. And this was the first demonstration, the first causal demonstration that the HERA model conceptualized on the basis of neuroimaging investigation was really working in our brain for at least for visospatial episodic memory tasks. This, of course, opened a series of other thousand studies, not only on memory, but also on many other cognitive functions. And another one that we did, again, together with Claudio, is to investigate also another model for episodic memory that appeared at the time. In this case, the model put forward by Roberto Cabezza was called Harold, that means hemispheric asymmetric reduction in older adults. Again, based on neuroimaging investigations, Roberto Cabezza showed that older adults, in order to maintain a good memory performance, had to reduce their asymmetries of activation. And both right and left prefrontal cortices were active during a successful memory task. While in young subjects, as you can see, the right prevalence during retrieval was still present. The neuroimaging derived model left open some very important questions. One is why this can happen. It could be a compensation mechanism, for example, helping to counteract the age-related cognitive decline, or the lack of asymmetries could be a dedifferentiation process, probably related with an age-related failure to allocate functional resources in a selective manner. And then this model, this Harold model, left open what actually happened to these functional asymmetries in encoding. So by using the same paradigm of stimulation that and the experimental setting that you saw before, we again tried to prove this model with TMS. And what we found was that in young adults, episodic retrieval depends primarily on the right prefrontal cortex, confirming our previous studies, whereas in older adults, it depends not only on the right, but also on the left prefrontal cortex, because also we produced recognition errors also when we went to stimulate the left dorsolateral prefrontal cortex, giving substance to the assumption that this activation are really working as a compensation rather than a dedifferentiation process. Instead, the functional asymmetries of the left prefrontal cortex in retrieval that we found in young subjects persisted across the lifespan, up to almost 8 years of age. So functional rearrangements for the mechanisms by which an old people can retrieve a memory, but according to this experimental setup, the same way of encoding of the information. So from these studies and from the other 1,000 studies on all cognitive functions that have been carried out with TMS, we can see now that TMS can add really something to neuroimaging investigations, because it overcomes the correlational aspects of EEG and functional MRI and positron emission tomography. It allows to establish a causal connection between the cerebral activity and behavior, not only concerning the stimulated area, but even for the cognitive process under study. So we can look at TMS as a technique able to obtain the functional resolution that closes the loop of the temporal resolution of EEG and of the spatial resolution of functional MRI. Another way to stimulate the brain that we studied a lot in these last 10 years is transcranial alternating current stimulation. This is a low intensity, almost imperceptible stimulation that we can apply on the scalp, on the desired regions of the scalp, provided by a battery, a 9-volt battery. And in this case, oscillatory currents can be given according to the endogenous rhythm. So we can apply a theta stimulation, alpha stimulation, beta stimulation, and gamma stimulation in the region that we want, in the network that we want, personalizing the way in which the current can reach the brain according to biophysical models that I will show you later on. How can transcranial alternating current stimulation works on our oscillatory activity? Conceptually, is very easy to understand. If we think, if look at this part, each one of these figurine is a neuron that is a neural oscillator with a proper vector of activity, okay? Quite anarchic activity, as you can see. The mean vector, all this vector is very small. But if I expose to a periodic external force as transcranial alternating current stimulation these neurons, all these neurons tend to have a phase alignment and an amplitude increase on their final cumulative activity. So we can entrain endogenous rhythm by using alternating current stimulation. There is some solid experimental evidence now that the currents that use of the human brain to entrain neural activity are physiologically effective. This is a recent study published on Science Advance in which these guys performed some recordings from 64 neocortical electrodes placed on the motor cortex of a monkey during stimulation at different intensity and 10 Hertz. When using intensities compatible with those that we are going to use in humans, a larger proportion of neurons of this region of the cortex can be entrained, and some entrainment could be seen even at a single neural level, okay? They also did some control stimulation because for excluding that such neural entrainment could not be due to some peripheral stimulation due to trigeminal afferent stimulation and so on. So with transcranial alternating current stimulation, we can modify brain rhythms in a very relatively easy way. And we know that different kinds of oscillatory activity accompany some certain cognitive tasks. For example, we know that alpha activity in sensory motor regions is linked with tactile perception. We know that theta activity in frontoparietal networks accompanies the brain that is trying to perform some working memory tasks. We know that beta activity is linked with movement. We know that gamma activity is accompanying tasks of selective attention and intelligence. So why to use transcranial alternating current stimulation to modify a rhythm? Because if a rhythm is associated with a performance in a certain task, if we are able, by applying external current at a given frequency, and this given frequency is able to increase the endogenous rhythm through the entrainment, then we could also modify the performance in the task under investigation. And this is what we did with Matteo Feurra and Emiliano Santarnecchi some years ago in our lab in Siena, published in journal Neuroscience. We verified in combining TMS and transcranial alternating current stimulation on the primary motor cortex in order to verify whether certain rhythm, induced rhythm by TMS, by changing endogenous rhythm, could modify the amount of corticospinal output. We used different frequencies of stimulation, theta, alpha, beta, gamma, on the motor cortex and on other regions. And we tested if something in terms of excitability was changed in the motor cortex itself. What we found is that just only when the primary motor cortex was stimulated at 20 Hertz, so at a beta rhythm, the amplitude of motor evoked potentials induced by the TMS given on the stimulating electrode were significantly increased. And nothing happened by using the other frequency. So the results of this study could represented the first evidence that the sensory motor 20 Hertz rhythm plays a specific functional role in increasing the corticospinal output, and that the effect, of course, was not artifactual because we used many experimental controls, and this effect was frequency dependent, and also had a certain regional specificity. Then, together with Emiliano Santarnecchi, we moved a bit forward, and we wanted to verify if gamma activity in the brain, inducing gamma activity in the brain, and in particular on the left prefrontal cortex, could somewhat improve some tasks, cognitive tasks related with fluid intelligence capabilities. Fluid intelligence is our part of intelligence that allows us to solve a problem taking apart our previous knowledge, okay? So we used the Raven matrices that you probably know, asking subjects, to 20 healthy subjects, to solve as faster as possible these problems that were of increasing complexity. The task was to find the solution among the different possibilities here to complete the series. The difficulty of the task progressively increased. There was one relation here, two relations, three relations, and in this case, tasks were of logic, contained logic constructs. We target this region, middle frontal gyrus, because previous neuroimaging investigations and lesional studies suggested that this was a likely candidate for being associated, whose activity could be associated with fluid intelligence tasks. And we again applied transcranial alternating current stimulation at different frequencies: theta, alpha, beta, gamma, and of course a placebo condition. What we found was that subjects were 15% faster in correctly solving only logic conditional trials, okay? And this happened only when the stimulation was given at 40 Hertz. That means in gamma range. And this was at the time, about 10 years ago, the first demonstration that gamma activity is really functional for solving, for helping to solve these logic trials in some way comparable to fluid intelligence tasks. Some colleagues in Oxford did not believe so much to our results. Professor Cohen-Kadosh asked to repeat this experiment in Oxford by using our experimental setup. We were a bit skeptical, but then we decided to agree, and the experiment was repeated on a large number of healthy subjects. And results were basically confirmed, and this was really a good news. But there was something that was unexpected appear. If we split the population of healthy subjects of Oxford students that participated in the study, we found that about 60% were called low performance, had a time of solution of these Raven matrices a bit slow, while 40% were really fast in fast performance in finding the solution to the tasks that you have seen. And you see that gamma stimulation of the left prefrontal cortex did not affect people, did not improve the performance of people that were already fast in solving those tasks, but significantly improved the performance in terms of time of people who were low performance. But among fast performance, about 25% of them actually worsened their performance in solving the task. And this was quite important because this study opened a sort of an ethical issue, because if these results are true, they could be a way to reduce cognitive inequalities among individuals. Now I see that we are going a bit long, so I have to shorten it by my presentation, and I would like to focus only on 2 or 3 points regarding the use of TMS as a neuromodulatory technique to treat some psychiatric conditions. Neuromodulation, what is? Neuromodulation means to deliver a small amount of electricity on a nervous structure area or hub or network, which should be part of or strictly connected with a dysfunctional network, with the objective to induce lasting changes in its function, thereby getting a therapeutic effect. This is what does, for example, deep brain stimulation for treating Parkinson's disease, but can be done also with non-invasive brain stimulation. Let's move. The potential of the therapeutic effect lies in the fact that applying trains of different kind of repetitive transcranial magnetic stimulation for several days, for several weeks, in a certain target region of the brain, can modify synaptic activity. And if we use an excitatory protocol, we can produce changes at a synaptic level that are similar to those experimentally observed for long-term potentiation, or LTP. And if we use protocols of intervention that are considered inhibitory at 1 Hertz or less, we can induce long-term depression in this synaptic in the synapses. So with TMS, we can restore a more physiological operating mode at a synaptic level, as if you think well, as every drug acting on the central nervous system is doing. But what is important for clinical application, that TMS can have local effects. Local means on the point of stimulation, but these effects can spread to the whole network which belongs to our cortical target. And this can be seen either neurophysiologically or by using neuroimaging technique addressing cerebral blood flow and metabolism. For example, in order to understand, to well focus this network effect, if by stimulating the primary motor cortex of a Parkinsonian patient that has deep brain electrodes within the subthalamic nuclei, a single TMS pulse can induce a suppression of oscillatory pathological activity in the subthalamic nucleus for several hundreds of milliseconds. And this is an elegant demonstration of this at distance effect. The International Federation of Clinical Neurophysiology has promoted a survey, an evidence-based survey, and now a clinical definite efficacy is obtained for depression, chronic neuropathic pain, and post-stroke motor hand recovery in the acute phase of a stroke. We have no time to go into these details, but what I would like to see is how we are moving in order to better found solutions for patients and more personalized approach. For example, we know now that the efficacy of TMS targets, in this case the left dorsolateral prefrontal cortex for treated depression, is related to the intrinsic functional connectivity with the subgenual cingulate cortex, which is part of the dysfunctional network in depressed patients. Or, for example, this is a study, a recent study from my lab, in patients with obsessive compulsive disorders, in a pilot study with nine patients, we found an improvement of symptoms of OCD domains by about 25%, which is not a little effect. By after 5 days of TMS of the supplementary motor area, and we found that connectivity changes in the network connecting with the targeted supplementary motor area with thalamus and basal ganglia, these connectivity changes were related to clinical improvement. But even that higher symptoms score, okay, correlated with higher connectivity, higher pathological connectivity between supplementary motor area and subcortical regions known to be included in the pathophysiological process underlying OCD symptom. This means that investigating connectivity might be a way to predict the clinical outcome of repetitive transcranial magnetic stimulation. The last thing, just 2 minutes to talk you about where we are moving now, thanks to Giulia Sprugnoli and Emiliano Santarnecchi, who is now a professor at Harvard University of New York. We are trying to use TDCS, which is another form of brain stimulation, which is based on direct currents rather than alternating currents, to modify brain perfusion. The idea moved from 2 studies in which either a nodal or a cathodal stimulation was able to modify brain perfusion in restricted regions of the brain, with a mechanism that is still a bit debated, maybe an effect on pericyte, on that are around vessels, or direct effect on endothelial cells, or even to some modification of activity of glial cells. But this is another story. We did this reasoning. If TDCS is able to modify perfusion, and if we have certain pathologies, I am referring to high-grade gliomas, superficial gliomas, which are characterized by important changes in local perfusion, then if we apply a personalized transcranial direct stimulation around the brain tumor, we could be able to change the tumoral perfusion. And this is an approach that outside of the brain is already known. There are many tumors of the abdomen or the liver that, especially in the north of Europe, are treated with intratumoral electrical stimulation that can produce necrosis of the internal part of the tumor. But no one has yet tried to verify what could happen with TDCS on brain tumors. So we did a pilot study with Giulia on 8 patients. We characterized the lesions in terms of necrotic core, of the solid part of the tumor, and of the surrounding edema. We developed a biophysical modeling taking into account the anisotropy of different tissues with the electrical stimulation by personalizing the type of stimulation on each single patient for the location of each single tumor. And what we found is that we got an overall decrease of intratumoral brain perfusion of about 36%, either in glioblastoma or in 2 cases of metastasis, without any effect of the surrounding edema on the necrotic core, on the ipsilateral healthy tissue, or on the contralateral healthy tissues. And this was obtained only with a single session of 20-minute stimulation performed inside the scanner. And this is a very promising results, in my opinion. Of course, it's a pilot study. We have no sham control, and we do not know which could be the long-term effects, especially if we use repetitive sessions of stimulation. Okay, Claudio, I guess I can finish here because the main message is given, and otherwise there is no time for.
I think that if you want extra 3-4 minutes, you can go ahead.
Okay. Regarding this last topic, we are moving now in this direction, thanks to Giulia Sprugnoli and Emiliano Santernecchi. Giulia did a wonderful study when she was in Boston with Dr. Golby at the Neurosurgical Center of Harvard, and looked at how the solid tumor was somewhat connected with the rest of the brain, and in particular in 2 resting state networks, ventral attention network and frontoparietal control network. Okay, and she was able to predict that the connectivity strength of the tumor cells within this network could be used as a biomarker for determining the survival of these people with, I would say, an impressive precision in terms of tenths of day. Of course, this opened some very interesting directions, because one, of course, is to use the tumor network connectivity as a survival biomarker once this approach will be validated across different laboratories. And another one is on the basis on our pilot study is to think to some personalized neuromodulatory interventions in order to, from one side, reduce the size of, reduce the intratumoral brain perfusions, but also trying to modify some tumors cells electrical properties that has been recently discovered, mainly in terms of connections with glial cell. So the idea is to perform daily sessions of transcranial direct current stimulations in order to reduce the size of the tumor and finally, maybe utopistically, trying to improve the long-term survival of these patients. So thank you very much, Claudio, for this opportunity, and I am open to questions, of course.
Thanks a lot for this fantastic presentation, showing not only the past and the present, but also the future of a non-invasive brain stimulation, brain connectivity, cognitive functions, but also therapeutical effects. So we had all in one presentation. Thanks a lot. So
maybe too much.
No, absolutely. We need this. We need this absolutely. And so I kindly ask the people connected to this presentation to write the questions in the chat. So I will read the questions to you for the discussion.
Okay.
To start, I want to your comments about the use of non-invasive brain stimulations with oscillations in order to get into the cortical networks, oscillatory cortical networks. You know that the cholinergic system, ascending dopaminergic system, or local glutamate, GABAergic circuits are very important to modulate the cortical oscillations. So my question is if with your kind of intrusion with oscillatory electrical stimulation or TMS stimulation, you can interact with neurochemical ascending systems.
Theoretically, yes. I guess that studies in this sense are really still needed. They are basically lacking. But what I can say you is that my co-worker Emiliano Santernecchi in Boston now is doing a clinical trial on a very attractive possibility, which is to use gamma stimulation, gamma transcranial alternating current stimulation, in order to modify the amount of beta amyloids in patients with Alzheimer's disease. And this clinical trial moves from experimental studies published in Nature 3 years ago by Iannacone et al., that in an optogenetic-based study demonstrated that interneuron of a mouse, of Alzheimer's disease mouse, if stimulated with light flickering in a gamma band, could reduce the load of beta amyloid, which is the pathogen protein of Alzheimer's disease, thanks to the effect of activation of certain glial cells and other kind of neurons. So I'm not sure to have answered to your question.
Yeah.
But the idea to really to go into the biology of the brain is apparent.
Yeah. It's a clear proof of concept. And from the audience, Ilaria kindly ask you if TMS can be applied to children in relation to the shape and size of the brain of these children. Yes. So there are cognitive studies or therapeutical procedures to
TMS can be used in children safely, and this is stated now in the new version of the international safety guidelines that appeared last week, so they are very, very fresh. And of course, when dealing with brain stimulation and children, we have always to balance possible risks and possible benefits. So there are certain conditions, for example, ADHD, that can benefit from brain stimulation. And in this case, I would say yes, but attaining, of course, to the practice, to the good practices of safety that have been already published in 2009. But the answer is yes, it can be used in children.
Okay, thank you, thank you. And one part of your talk was about the existence of different oscillatory activity in different networks with different frequencies and impact on cognitive processes. So, do you think that it's possible to combine different oscillatory stimulations simultaneously in different parts according to the intrinsic frequency of the cortical area stimulated?
Yes, this is feasible, and there is some group around the world that is trying to do this. For example, in the memory domain, the idea is that memory is a complex phenomenon, and can be accompanied by rights, for example, in the theta band. But we know that also some gamma activity in certain brain regions can be really crucial for a good memory performance. And there is the possibility to combine a transcranial alternating current stimulation that has a theta rhythm on an underlying theta rhythm, and from time to time, on the negative or positive peak of oscillation, there is a gamma burst. It theoretically is possible. Clearly, in my opinion, even a so complex stimulation does not satisfy the full complexity in terms of oscillation of our brain. But, science research is incremental, and maybe we can we can arrive closer to what really happens in terms of oscillation inside the brain.
In a very strong message of your presentation is about the possibility that modulating the activity of the cortical neurons, you can produce some probably neurohormonal local effects on healthy of the neurons and tumoral processes, also by means of neurovascular effects and so on. So my point is, if with your non-invasive brain stimulation, you can change the efficacy of the cortical networks, but not the real evolution of the disease when it's based on neurobiological failure, these homeostatic processes. Can you really revert this neurobiological basis of the diseases?
The answer is not yet, in the sense that, if we are targeting Alzheimer's disease, for example, we are in front of neurodegenerative disorders that proceeds more and more in time, and every kind of non-invasive brain stimulation intervention that we can do is limited in time. And I really do not expect that some new neurons will reappear in a deep brain. What we can do is one way is the one of reducing beta amyloid, which is not okay, which could be a good objective. The other way is helping to somewhat relying on the neurons that are still functioning in a brain in order to compensate for the loss of function due to neurodegeneration. I guess that more than this is difficult to think at this stage. This is what is already done, for example, with other invasive techniques as deep brain stimulation. Deep brain stimulation in Parkinson's disease does not slow the neurodegenerative process of the disease, but improves dramatically mainly motor symptoms, and sometimes also no motor ones. But you have electrodes in the brain that are working 24 hours a day, which is not feasible with it's not sustainable with non-invasive brain stimulation, unless one takes a stimulating helmet, bring it at home, and and uses it in a domestic context. But this opens another series of problems.
Okay, I read your book. I liked very much your story. It's a really nice biographical narrative, stripped together with emerging hot topics in neuroscience.
You are also included in my book.
Yeah. Minimally, you are right, because really your network was really extended over time. But my point is, what's the core message of this book? The really strong seminal take-home message
of the book?
Yeah.
The message is that I hope that people will enjoy reading this book, finding and if the reader is already experienced in neuroscience, will probably find some new point of view of how to approach the brain. If the reader is curious of the brain, but is not involved in the field, I guess that it could be catched by some stories that I'm talking about, that are also sometimes funny, sometimes not funny, but still real life stories and stories in the lab that can, however, make the reading the book enjoying as well. So it's a halfway between a scientific essay and something that is not scientific. The microphone. Your microphone is switching off.
Yeah, you are right. Thanks a lot for your discussion at the level of the presentation.
And thank you everybody, to all the attendees.
Okay, so bye to you, and thanks to all people connected to us. We have more than 60 people following your seminar, so we are happy of this service. This is one of the seminars of Sapienza Neuroscience, a network of neuroscientists in Sapienza University of Rome, working together and organizing these international seminars. Thanks a lot. Bye bye.
Thank you so much, Claudio. Ciao, ciao a tutti. Ciao.